Hello everyone, this is Dr. Govindra Garg. In this video, we will discuss two important topics. One is clinical trials and second is pharmacovigilance. So after the video, I will want that you should know about these important things. First, what are the clinical trials, their main importance, what are the various phases of clinical trials, then the concept of placebo and blinding in the clinical trials, some salient features of drugs and cosmetic act which are important relevant to MCQs, adverse drug reactions, types of adverse drug reactions, and lastly pharmacovigilance. So we'll be covering all these topics in this video. So we'll start our discussion. Currently we have thousands of drugs in the market. The question is how they came into the market. Suppose I develop a drug in my laboratory. I am working in pharmacology department. I develop a drug, and according to me, this drug is a very good antihypertensive drug. So I made 10,000 tablet of that drug. Go to pharmacist and say him that please sell this drug. Can I do that? Obviously not, because drug is a chemical substance. It can cause any harm. If someone is killed, who will be responsible? So drugs cannot be sold without proper testing. So first of all, we have to test the drug for the safety as well as for the efficacy, whether it is effective or not. So when we are sure, only then the drug can come into market. So we need to test the drug. First of all, the drug is tested in the animals, then the drug is tested in the humans, and only after that the drug can come into market. So this testing of drug in humans is called as clinical trials. Now, before discussing about clinical trials, first we will test the drug in animals. The study of drug in animals, it is known as, there is a committee known as Committee for the Purpose of Control and Supervision of Experiments on Animals. Now this is a committee which is known as CPCSEA. So this committee gives the guidelines how we can test the drug in an animal. Obviously, animal should not be unduly harmed. So there must be some proper objective, and without harming the animal too much, we should use the drug in the animals. Okay. So the guidelines which we follow for animal studies are called as CPCSEA guidelines. Okay. The animal studies are called as preclinical studies. Remember, whenever we write the word clinical, it means on humans. Okay? So first of all, we will test the drug in animals. So basically, we will test two things in the animals. First, whether the drug is effective or not. So how we will test? We have various animal models. Like if we want to test a new drug, whether it is effective anti-diabetic or not. So first of all, animal should be diabetic. So we will make an animal diabetic first. Like we can remove the pancreas of the animal, or we can give a drug called as streptozocin, which will destroy the pancreas of the animal. So the animal will become diabetic. Then we will give the drug and see whether it is controlling the blood sugar or not. Okay. So there are animal models for almost every human disease. Second thing we need to test is this safety, whether the drug is safe or not. So what we will do after giving the drug to the animals, what we will do, we will do all the test on the animal. We will take the blood sample of the animal. We will do hemogram, liver function test, kidney function test, all the tests we will do. And after that, we will sacrifice the animals, pick up all the organs of the animals and do the histopathological studies whether there is any harm on any organ or not. So we can come to know the safety of a drug. So first of all, we test the drug in preclinical studies. They are not called trials. Remember, preclinical are called as studies. So in preclinical studies on the animals, under the CPCSEA guidelines, we test the drug. Now, if the drug is found to be safe and effective in animals, okay, then we can think that this drug can be good for humans. So I have tested the drug in the animals. Now I made 10,000 tablets and go to pharmacist that please sell this drug. Can I do now? No, the answer is still no, because we have tested the drug in the animals only, but we have to give it to humans. Okay? So first of all, we have to test the drug in humans also, and that testing of drug in humans is called as clinical trials. But before starting the clinical trial, there must be some authority. There must be some licensing authority who has the power to say that yes, this drug is useful, you can use, this drug you cannot use. So there must be some authority. So that authority or licensing authority in America, it is known as FDA, United States Food and Drug Administration. Whereas in India, the similar authority is Central Drug Standards Control Organization. The name of the organization is Central Drug Standard Control Organization, and it is headed by Drug Controller General of India. So, organization name is Central Drug Standard Control Organization, whereas the authority or the person who is head of this is Drug Controller General of India. Okay? So, in America it is FDA, in India it is DCGI. They have the power to give the license for the drugs. Okay, so now we want to test the drug in the clinical trials. We want to start the clinical trials, but for that also we require permission of licensing authority. If we want to market the drug later on in only India, then we will require the permission of DCGI. But if we want to market the drug worldwide, we will want permission of FDA. Okay, so first of all, we will write an application along with the animal data. We will send that application to the licensing authority, and in that application we are requesting for starting the clinical trial. Okay, and the name of that application is INDA. The application name is Investigational New Drug Application. So what we are asking in this application is, we are sending the animal data and we are asking that we have tested in the animals, and according to us the drug can be good for humans. So please give us the permission to start the clinical trials. Okay, so clinical trials can be started only after we get the permission from the FDA or DCGI for the INDA. So INDA, when it is approved, only then we can start the clinical trials. Okay, so when it is approved, now we can do clinical trials, which are done under 4 phases. We have 4 phases of clinical trials: phase 1, 2, 3 and 4. So obviously the first trial in humans is phase 1 trial. Now this trial is done in healthy volunteers. So one very important thing to understand, this is done in healthy volunteers. The question is, why to test in healthy person? Because we have to give a drug to a diseased person. Then what's the point in testing in a healthy person? The reason is simple. Diseased person is already having compromised body. Okay? So thus, if a drug causes some side effect, that may not be tolerated by them. Whereas a healthy person has a good immune system. So even if some side effect occur, the body can manage better. So based on this theory, mostly we will test the drug in healthy people first. Okay, but some drugs are likely to be very toxic, like anti-cancer drug. So if we give an anti-cancer drug to a healthy person, even that person can develop bone marrow suppression or other severe side effects. So it will be unethical to test such a toxic drug in a healthy person. So for such type of drugs which are known to be very toxic, we can directly test the drug in patients also. Okay? So phase 1 is normally done in healthy volunteers, but in rare circumstances we can do them in patients also. Okay, now we are testing in healthy volunteers. So what we will do? Can we do the safety or can we do the efficacy? My first question. Or in simple language, I can ask, what cannot be done in phase 1 trials? The answer is efficacy. Efficacy cannot be tested in phase 1. The simple reason is, we are giving the drug to healthy person. Healthy person do not have the disease. So when we give a drug, how we will come to know whether the disease has been treated or not? Like suppose we are making some anti-tubercular drug and the person is not having TB. So how we will come to know whether it can treat the TB or not? So efficacy cannot be tested in phase 1. That is important thing to remember. So when we cannot test the efficacy, what is left? Yes, we can test the safety. But anything more important than safety? Asking the same question in different format. before phase 1 trials, how many humans have received this drug? How many humans? Zero. We have not tested in humans. We are giving only to the animals till now. So when we have never given a drug to any human, how we will come to know what dose we need to give? So that we will find out in phase 1 studies. Although from the animal data, we can apply some formulas and calculate the human dose. But that dose is not a single value, that is comes in a range. So that is known as human equivalent dose. So we will calculate human equivalent dose from the animals. So like if we are using this much in the mice, this much we need to give in humans. If we are using this much in the rabbit, we need to give this much in the human. So we can calculate a human equivalent dose from the animal studies. Like the dose comes from the 50 milligram to 500 milligram. This range has come. Now whether we need to give 50 milligram or we need to give 500 milligram, how we will come to know? That we will test in phase 1 trials. So what we will do basically is we will call one person and we will give him very small dose. That means the smallest of the human equivalent dose. Like if the example we are saying is the dose comes in a range of 50 to 500 milligram. So we will call one person and give him 50 milligram. After giving 50 milligram, we will do all the tests. We will do hemogram, LFT, KFT, coagulation profile, we will do ultrasound, CT scan, whatever test is possible, we will do. Every test we will do. And suppose there is no abnormality in any test and the person is absolutely fine. So we will send that person home. We will call next person, give him higher dose, 100 milligram. Again, do all the tests. That person is also good. We send him home. Call the another person, give all higher dose. So we will keep on increasing the dose and we will stop at that point where the side effects are intolerable. Okay. So that dose we can calculate, which is known as maximum tolerable dose. Maximum tolerable dose. So that is the major aim of phase 1 clinical trials, to calculate the maximum tolerable dose. Okay. So in the MCQs, many times MTD is not in the option. So how you need to answer, what is the major aim of phase 1 clinical trials? So if they write MTD, that is the best answer. If maximum tolerable dose is not in the options, then you should mark that we will do it for dosing, because we are calculating the maximum tolerable dose. And if that is also not given, mark answer as pharmacokinetics, because the dosing is related to pharmacokinetics. And if that is also not given in the option, then mark the last answer as safety. So in this sequence you need to answer what is the aim of phase 1 clinical trials. Okay. And do not mark efficacy, because we cannot know efficacy in phase 1 clinical trials. Okay. So we have done phase 1 trial. After phase 1 trial, what answer we get is do not give more than this milligram. Okay, so we get the range that in further clinical trials, the dose will always be less than MTD. Okay, so we have calculated the dose and now we go to phase 2 trial. Now phase 2 trials are the first trials in the patients. First trials in the patients, we take small number of patients. Basically, we will go to a one hospital, and whatever number of patients we get from that hospital, maybe 50, maybe 80, maybe 200, maybe 300. So whatever number of patients we get, we will enroll them. Okay. So usually the range we say is around 50 to 500. Usually 100 to 500, 100 to 200, whatever number of patients we get. Okay. So few hundred patients we will enroll. And what we will see here, we will give them the drug and we'll see whether the drug is effective or not, whether the drug is safe or not. So we can test both of them. So what is important to remember is phase 2 trials are the first phase in which we can test the efficacy of a drug, because in phase 1 we cannot know the efficacy. So if the MCQ asked in which phase we first of all can test the efficacy of a drug, yes, it is phase 2 trial. Okay. So apart from efficacy, side by side we will also note safety. If any different adverse effect has come, we can note that down. So phase 2 trials, first time efficacy is done. Okay. Now after phase 2, we have confirmed that the drug is safe, the drug is efficacious. Now I have made 10,000 tablets again and I go to the pharmacist I have tested the drug in the humans also. Now please sell the drug. Can the person sell now? The answer is still no. Why? Because we have tested only in 100 to 100 persons. And we are not 100% sure whether the drug will be effective in everyone. So we first must confirm the result of phase 2, and that confirmation is done in phase 3. So phase 3 is nothing but everything like to phase 2. That means it is done in patients, and we will do the same thing, safety and efficacy. But the difference is this is done in large number of patients, thousands of patients, usually 2000, 3000. We take up to 5000 patients. Thousands of patients are taken. Okay. And second thing, these patients are enrolled from various centers. They're not from one place. Like phase 2 was only from one place. Phase 3 are from different places. Some from Delhi, some from Bangalore, some from Hyderabad, some from Kolkata, all over the country. And sometime across many countries also. So first question, why we have enrolled so many patients? Because we want to confirm the results. If a drug is found to be safe and efficacious in 5000 people, most likely it will be efficacious. So phase 3 will confirm the result because we are testing in so many patients. And number 2, why we are taking from different centers? Because we want to cover every genetic makeup. Sometimes that happen is a drug may be working only at one area, not at other place. So we want to generalize the results. Okay. So phase 3 will confirm the results of phase 2. So read carefully. If the question asked in which phase we confirm the efficacy, answer will be phase 3. In which phase we first of all do the efficacy, it is phase 2. Okay. So after phase 3, now we are sure that the drug is safe and efficacious. Okay. So after phase 3, I made 10,000 tablets. Again, I went to pharmacist that I have developed the drug. I have confirmed that it is safe and efficacious. Now please sell the drug. Can the person do? Still answer is no. Why? Because we may know very well how to drive. But if we do not have driving license, we will get challaned. So now what we need to do? We need to get the license. For getting the license, what we will do? We will again remember the licensing authority. So that means FDA or DCGI, we will write them another application, and that application is now known as new drug application or NDA. So after phase 3, we will write new drug application. Along with this application, we will send data of phase 1, 2 and 3. Okay. Now do you remember first application which we had written to FDA was INDA. So what is the difference between that application INDA and this application NDA? I is absent. That was INDA, this is NDA. So that means the drug is no more investigational. It has already been investigated into and it has become a new drug. So what we do is in this application, we are asking the permission to market the drug. So we want that the drug we have already tested, we have spent a lot of money. So now the drug should come into market and we should earn some money. Okay. So after phase 3, we will write a new drug application. FDA will study the data of phase 1, 2 and 3. And if they are convinced that the drug is good for humans, then they will give the permission for marketing of a drug. So after phase 3, the drug will come into market. Yeah. But what they will note particularly is they will note that whether the studies have been done under GCP guidelines. So FDA has provided the guidelines which are called as good clinical practice guidelines. So basically these guidelines are given, GCP guidelines are given so that the data which is generated from phase 1, 2 and 3, it is more accurate and more credible. Plus the patient should not be unduly harmed. So basically to keep the interest of the patients in mind, as well as to make the data credible, we are giving some guidelines. So if the study is deviating from these guidelines, then the FDA will not approve the drug. Okay. So after phase 3, after approval of NDA, the drug will come into market. But still we need to do these studies which are called phase 4 studies. And phase 4 studies are also known as post marketing studies. post-marketing studies. Now the question is, we have already tested the drugs in thousands of patients. We have done phase 1, phase 2, phase 3. We have confirmed the results also. Now why to test now? The drug is already in the market. Patients are already getting the drug. Why to test now? The reason is, phase 3, they are done for mainly rare side effects of a drug. Like we can take an example. You all know about a drug called as aspirin. Yes. Aspirin is a very good anti-inflammatory drug. Everyone knows. But it has one major side effect, that it can cause peptic ulcer disease. In many patients on long-term use, it can cause peptic ulcer. So then we developed a new drug which is called as rofecoxib. Rofecoxib is again very good anti-inflammatory drug like aspirin. But it is not causing peptic ulcer. So when we find this, that it is as good anti-inflammatory as aspirin, but it is not causing the major side effect of peptic ulcer, then we started developing rofecoxib. And we did the clinical trials. We did phase 1, phase 2, phase 3 clinical trials. So all the clinical trials confirmed that rofecoxib is good anti-inflammatory drug and it do not cause peptic ulcer disease. And when the data was submitted to FDA, it was approved. So rofecoxib, it came into market. Now when it came into market, all the doctors threw aspirin into dustbin, that this is a useless drug. Rofecoxib is the main drug. So within 2-3 years, rofecoxib was given to billions of people. Okay? Billions of people received this drug. And when we did the post-marketing studies, those studies also confirmed that it is a good anti-inflammatory drug and it do not cause peptic ulcer. But the studies in the phase 4 post-marketing studies, they revealed that it is not causing peptic ulcer, but it is causing myocardial infarction. Now the question is, it is causing MI. Why we did not come to know in phase 1 or phase 2 studies or phase 3 studies? Because it is causing 1 MI out of 50,000 people when we give. And if you remember, maximum number of patients we have enrolled in phase 3 trials were 5,000. So maybe that patient is not there. So that does not mean that it cannot cause a side effect. Yeah? So, but when it is given to billions of people, several thousands can develop MI. Yeah? So when we came to know that rofecoxib is causing MI, so we thought that MI se अच्छा तो peptic ulcer ही. So rofecoxib was withdrawn from the market. So this type of side effects, which are seen in very few patients, they are called as rare side effects. And the rare side effects we can come to know only from phase 4 study. So the major aim of phase 4 studies are rare side effects of a drug. Okay. And second important is long-term side effects. So some drugs, like we know steroids, they can cause osteoporosis, but that will be seen when it is used for 4, 5, 7 years. But all the clinical trials are time-bound studies. Remember, we are spending a lot of money on them. They cannot be unlimited. Yeah? They may be for 2 months, 6 months, 1 year. So if a drug is causing side effect after many years, we cannot know in phase 1, 2 or 3. But when the drug is in the market, we can continue the study for many years. So we can come to know about long-term side effects of a drug also. Clear? So rare side effects and long-term side effects of a drug can be known only in phase 4 trials. Clear? So these are the 4 phases of clinical trials: phase 1, phase 2, phase 3 and phase 4. Okay. Now in between, one more clinical trial was formed, which is called as phase 0 clinical trial. Now something important to remember, first of all, it is clinical trial. Remember, the word is clinical, and trial means it will be done in humans. So it is not in animals. So you need to remember, phase 0 is done in humans. Now the question is, why phase 0? Now it has been found that when we are developing a drug in clinical trials, out of thousands of the drug, hardly 1 or 2 drug is successful, many other are failed. Okay? But the money is spent on every drug. Okay? For developing one clinically important approved FDA approved drug, a pharmaceutical company spend billions of dollars. Okay? and the cost is increased because many drugs fail. Now, pharmaceutical companies want to know that whether a drug has high chances of being successful or low chance of being successful. So if they beforehand come to know that this drug has high chance of becoming successful, they will develop that drug only. Okay? So how they will come to know? So they tried to find out why a drug can fail. And they came to the conclusion that two major reasons why majority of the drugs fail in clinical trials are: number one, drug is not going to that area where we want it to work. So that means it will not be effective enough. Second reason can be the drug is concentrating in some area where we do not want. So that can lead to toxicity in that organ. So the pharmaceutical companies thought that if beforehand they come to know that which drug is going at which place, so they can choose which drug to develop and which drug not to develop. So for that they developed a separate study known as phase 0 clinical trial. The other name of this is micro dosing study. Phase 0 are called as micro dosing studies. So what we do, as the name tells, we will give a drug in very small dose. The dose usually given is less than 100 microgram. 100 microgram, micro dosing, or 1/100th of the human dose. Like if we want to give in humans as 100 milligram, it should be 100 microgram. Remember, 1/100th is 1 milligram, but maximum out of these two. So either 100 microgram or 1/100th of the human dose, whichever is lesser. We will not give more than that. Okay. Now, if we are giving a drug in so low dose, will you get any therapeutic effect? The answer is no. So in phase 0, we cannot know the efficacy of it. Now, do you expect that with so low dose there will be side effects? Obviously, we cannot. So we cannot know the safety of a drug. So we cannot know the efficacy, we cannot know the safety. Then why we are doing? What we do is, to this small amount of a drug, we will attach a radioligand. So after attaching the radioligand, we will give the drug to the humans, and then we will monitor on the counters. We will sit on the counter and see where all the drug is going. So we can come to know that where all the drug is getting concentrated. Clear? So if a drug is concentrating at the desirable place and not concentrating at the undesirable place, only then we will start developing this drug. Yeah. So what we can do in phase 0 study is, we can see the pharmacokinetics of a drug, or we can see the movement of drug in the body. Okay, so that is phase 0 clinical trial. Most important thing to remember, it is not mandatory. FDA do not say that you have to do. Okay, and it is done in humans. We cannot do efficacy, we cannot do safety, but we can do the pharmacokinetics, or we can see the movement of drug in the body. Okay. So we discussed about various phases of clinical trials. So quickly we will answer the MCQs. We will revise basically. So which is the first phase in humans? It is phase 1 trials. Second, in which phase we first of all know efficacy of a drug? Yes, it is phase 2 trial. In which trial we cannot know the efficacy of a drug? Yes, it is phase 1 trial. Which trial contains maximum number of patients? Yes, no, it is not phase 3, it is phase 4. Remember, phase 3 has up to 5,000, phase 4 unlimited. Now, which trial can be done after approval of IND? It is phase 1. Then, after which phase you can file NDA, new drug application? After which phase? It is done after phase 3. Which is the post-marketing studies phase? It is phase 4. After which phase the drug can be marketed? It is phase 3. After, if they ask, it is phase 3. If they ask which phase is post-marketing, it is phase 4. Now the question which we have not covered here, which phase has maximum risk of failure? Most of the drugs fail in which phase? So we can see what we do in phase 1. We are calculating the dose, maximum tolerable dose. It may not be 100, it may be 50. Okay? So there is very low risk of failure because we are just seeing the dose. Okay. In phase 2, we are first of all saying about efficacy, whether that drug is efficacious or not. High chance that it may fail because we are doing first time. Okay. Now, for which drugs we do phase 3? Which have passed phase 2. If the phase 2 has already been passed, high chance that it can be passed in phase 3 also. And phase 4, we know rarely the drug fails, like rofecoxib, they are very rare occurrences. Okay. So maximum risk of failure is in phase 2 of clinical trials. Okay. And which phase is microdosing studies? Yes, it is phase 0. Okay. So these are some of the important points regarding the different phases of clinical trials. Okay. Now, in the clinical trials, there are two important terminology. One is placebo, second is blinding. Okay. So we'll try to understand what is the meaning of this. Suppose you want to test a new painkiller drug, whether it is stopping the pain or not. You want to know that. So you give this drug to 10 people. You give this drug to 10 people, and after giving the drug, you will ask whether the pain is relieved or not. Obviously. Yeah. Now, suppose after giving the drug to 10 people, 5 people say that the pain is relieved, and 5 people say that pain is not relieved enough. Now, is the drug effective or not? Or if 7 people say that the pain is not relieved, 3 people say that the pain is relieved. You cannot say, because pain is a subjective sensation. Sometimes just the feeling that I have received a drug may relieve the pain. This you will see when you go to internship. You are on a duty in the night, and one patient is again and again complaining that, doctor, I have a pain, I have pain, I have pain. And you are intern, you do not know which drug to give, which drug not to give. So you say, sister, give him a normal saline injection. So sister give an injection of normal saline, and with 10 minutes, 50% of the patients say that, doctor, now the pain is relieved. Okay. So that effect is known as placebo effect. Okay. So what is important is we have to compare this new drug with something, and with which we will compare it with placebo. So what we will do is, we will not give the drug to 10 patients, we will give to 20. To 10 patients we give the new drug, to 10 we will give the placebo. Placebo is dummy medication. Placebo is dummy medication. That means everything is like drug. Same shape, same size, same smell, everything is same. The only difference is it do not contain the drug. Okay. So it is a dummy medication, sugar coated thing. Okay. So to 10 people we will give the drug, to 10 people we will give the placebo. Suppose in the placebo group, 5 patients are saying pain is relieved, 5 says do not, and same happen with the new drug, 5 says relieved, 5 says not relieved. That means that is placebo effect only. Drug is not effective. But suppose in the placebo group, 8 patients say that pain is not relieved, 2 says pain is relieved. Whereas in drug group, 2 says pain is not relieved, 8 says pain is relieved. So now the chances that the drug is working. Okay. So we have to compare it with some standard that is known as placebo. Placebo means dummy medication. Okay. Now, if we say that 2 patients have got pain relief by placebo, how it can happen? It is assumed that when we give anything to a person, that means placebo to a person's body may secrete endorphins. In the brain, there is secretion of endorphins, and endorphins may be responsible for the final effect of a drug. They are endogenous opioid system. Okay. So placebo may work in some people. Obviously, do not work in everyone, but in some people, placebo may work. Okay. So it is basically a dummy medication. Okay. Now suppose we have taken 20 people. To 10 we have given a new drug, and to 10 we have given the placebo. But we have told everyone that you are receiving the drug and you are receiving placebo. So if we have said that, then that uh, effect which we want, the feeling that I've received the drug, that will go away. So there is no use of doing that, because placebo effect will not come. If I know that I am getting normal saline, I'm not getting a drug, so obviously my placebo effect will go away. So what we need to do is, we need to make the subjects unaware, that they do not know whether I am getting a drug or I am getting a placebo. So that is known as blinding. So blinding means unawareness. Blinding means unawareness. The subjects do not know which thing they are receiving, whether they are receiving the drug or they are receiving the placebo. Okay, so now important thing to understand here is there are two things. One, we say patient, second we say doctor. But in clinical trials the terminology is different. We do not say patient. In the patient in the clinical trial is known as subject, whereas we do not say treating doctor, we say investigator, because whichever is doing the investigation we will say that is investigator. Clear? So subject means who are receiving the drug, and investigator who is giving the drug. Okay, so there are two major type of blinding. One is known as single blind and second is known as double blind studies. Now what happens in single blind study is only the subjects are unaware of the treatment. That means we are giving to 20, but none of those 20 know who is getting a drug, who is getting a placebo. But the investigator know who is getting placebo, who is getting the drug. Okay. Whereas in double blind studies, both the subject as well as investigator are blind. That means neither the subjects know nor the investigator know. So this will remove the investigator bias. So many times there is bias generated because I know that this person is getting a drug, so this person should get the effect. Where this person is not getting the drug, this person should not get the effect. So that bias can come. So that investigator bias can be removed by double blind studies. And double blind studies are considered to be the best clinical studies. So double blind studies are done when we want to confirm the results, and that is in phase 3. So phase 3 studies are usually double blind randomized controlled trials, whereas single blind studies are usually done in phase 2. Okay. Whereas phase 1 is usually open label studies in which no blinding is done. Open label means no blinding is done. Okay, so blinding means the person is unaware of treatment. Single blind means only subjects are unaware. Double blind means both subject and investigator are blind. Okay, now is a term also known as triple blind studies. Although this is not of much significance, in triple blind studies, apart from subject and investigator, even the statistician is unaware. That means who will analyze the data, even he do not know which is getting the drug, which is getting the placebo. Okay, so best studies are phase 2 trial, phase 3 trials in which there are double blind studies. Okay, so that is about the blinding and placebo. Now one more thing, sometimes many people get confused between another term, excipients. So what is the difference between placebo and excipients? They are totally different things. Placebo, we say it is a dummy medication, whereas excipients are the substances which are added to the drug. Normally, suppose we are making aspirin or we are making any drug. Many times the drugs are not stable. To increase the stability, we have to add the inactive substances. Or sometimes the taste is very sour or very bad. So to mask the taste, we need to add some excipients. Flavoring agents we may need to add. Clear? So in the drug, any substance which is not active, which are used to modify some other property, they are called as excipients. Okay, so that is placebo and blinding. Now we will move to, there is a law in India which is known as Drugs and Cosmetics Act. Yeah? So although there is a full law, but we will not go into details of that law. We need to know two important things for MCQs. One, two schedules we need to know. And second, we need to know about some named drugs, different type of drugs which are named under this law. The first thing, schedule, you need to know two schedules. One is Schedule H and second is Schedule X. Schedule H means these are those drugs which require prescription. Means these drugs should not be prescribed without taking the prescription from the patient. So only those drugs which require prescription, they are called as Schedule H drugs. Whereas if we do not require prescription, those drugs are called as OTC, over the counter drug. That means any person can go and say that give me this, the pharmacist will give. But those drugs which need prescription, they are called Schedule H drugs. Whereas Schedule X drugs are mainly for addictive drugs. addictive drugs. So basically in Schedule X, what happens is, apart from getting the prescription, obviously prescription is required. Apart from prescription, the pharmacist has to keep the photocopy of the prescription for minimum 2 years. So this is the regulation for pharmacist. They need to keep the prescription with him for 2 years. Only then the person can prescribe the drug. Okay? So highly addictive drugs like ketamine, amphetamine, they are coming under this category, Schedule X. And again revise, Schedule X and Category X are not same thing. Category X is according to pregnancy safety. Category X are teratogenic drugs like thalidomide. Okay, so thalidomide is not Schedule X, it is Category X. Okay, so that is about schedules. You should know Schedule H is prescription only drug and Schedule X is for addictive drugs. Okay, then second thing, there are some drugs which are named in this schedule, like 3 terms you need to know. One is spurious drugs, second is misbranded drugs, and third is contaminated drugs. Now what is the meaning of these are spurious drugs? Spurious drugs basically are those drugs where a new pharmaceutical company, like I have opened a pharmaceutical company 2 months back and no one in my country know about that my company. So if I make a drug, there is very low chance that the people will believe me and take my drug. So what I do is I will try to mimic the drug with some reputed company like Ranbaxy, Cipla, which everyone knows. So I make a drug in such a manner that people confuse with that. I keep the similar name or exact similar packaging and everything like similar. So that type of drug is known as spurious drug. So spurious drug simply means that an unknown or less reputed company is trying to take the advantage of name of a reputed company. Okay. Then misbranded drugs are those drugs where is anything wrong written on the label. Like on the drug label, we want that it should be written that it is for Schedule H, that means only for prescription only drug. If that is not written, it will be misbranded. If on the drug label it is written that it contains 100 milligram paracetamol, but actually it contains 50 milligram only, it is misbranded. So anything which is wrongly written on the drug label, that drug is known as misbranded. Okay. Then contaminated, from the name you can remember, you can find out this drug contains some filthy material or unhygienic material. Like very commonly the question is asked, they will give a tablet from which a hair is coming out. So which type of drug it is? So it is contaminated drug. So you need to know only about these things. Okay. Now we will move to our next topic, which is adverse drug reactions. Now adverse drug reaction is anything adverse caused by drug. So what is basic important thing to remember here is adverse drug reaction. You need to remember two terminology. One is adverse event, second is adverse drug reaction. Two things. Adverse event means anything wrong which happens after person take the drug, whether it is caused by a drug or not caused by a drug. All of them are called adverse events. Whereas adverse drug reaction means those adverse events which are caused by a drug. Okay? So among these 10 adverse events, whichever are caused by a drug, they will be called as adverse drug reactions. We will discuss these again little while later. But here we will discuss that adverse drug reactions can be of many types. So we can divide them into various types like type A, type B, type C, type D, type E, and type F. Different type of adverse drug reactions. Now type A means these are augmented. Augmented means the normal pharmacological action is augmented, increased. Okay? So what happened that we know that the drug is causing this because of its pharmacology. Okay? But when it is caused more than required, that is called as adverse effect. Like two type of augmented reactions are there. First is known as toxic effect. Like we know that the drug is antihypertensive, so it will decrease the BP. But if we give an overdose, it causes hypotension. So that effect is known as toxic effect. So toxic effect is basically the normal pharmacological action which we want for the person. We want to reduce the BP, but that is exaggerated. Clear? Then second type are called as side effect. Now this action we know that the drug is causing, but we are not using for this. Like atropine, we know atropine it is causing mydriasis. So we are using the drug in the eye to produce mydriasis. But at the same point, we know that atropine can cause dry mouth also. So we are using it for mydriasis, but it is causing dry mouth. That is a side effect. It is a normal pharmacological action, but we are not using it for this purpose. So it is a side effect. Okay. So toxic effect and side effect, but both of them, the special thing is these are predictable, because from normal pharmacology we can predict that these can happen, and these are usually dose related. If we increase the dose, more side effect will be seen. So dose dependent and predictable side effects are type A side effects. Okay. Then type A ADRs are known as bizarre. So type B is bizarre. B for bizarre. Bizarre means unpredictable. Unpredictable. We cannot know whether they will happen or they will not happen. Very simple example here is allergy. We do not know which person will develop allergy to that drug. Okay, so they are bizarre. Then C means chronic. So as the name says, these side effects will occur when we use the drug for many years, very long time. Like we know steroids cause osteoporosis. So steroid will cause osteoporosis when we use them for many years. So this type of side effects are chronic. Then D for delayed. Delayed means we have given the drug now and side effect will occur later on. So one of the very important delayed side effects are teratogenic effects. We have given the drug now to the pregnant female, and after 9 months the baby will suffer from some malformation. So these are called delayed side effects. Then E is end of dose. End of dose side effects, or simply withdrawal effects. So till the time we keep on giving a drug, it's okay, but when we stop the drug, the side effect starts. Like antidepressant drugs, we keep on giving antidepressant drugs, they control the depression, but whenever we stop, they cause the side effects known as discontinuation syndrome. Okay. Then F means failure. Failure of therapy means the drug is not able to treat the disease for which we are using. Okay. Now this is very important. Thus failure of therapy, it is considered to be an adverse drug reaction by FDA. FDA consider it to be adverse drug reaction, but WHO do not consider failure to be adverse drug reaction. So this is the simple reason why these organizations have been made. Food and Drug Administration, the purpose is to approve the drugs. So if it has approved a drug and that drug is not doing its work, according to him it is adverse. So they consider it as an adverse drug reaction. Whereas the function of WHO is to give guidelines for health. They are saying that if this is not working, use some other drug. So they do not consider it as adverse. Okay. So if a drug failed to produce the action, FDA consider that as an adverse drug reaction. Okay. So these are the different type of adverse drug reactions. Okay. So now we can move to our next topic, which is pharmacovigilance. So pharmacovigilance, from the name you can see, we have pharma, we have vigilance. Pharma means drugs, vigilance means to keep an eye. Okay? So why we want to keep an eye on any person? We want to keep an eye on any person so that he should not do anything wrong. Okay? So here we are keeping an eye on the drugs so that they should not do any wrong. And what is the wrong thing caused by drug known as? Yes, it is called as adverse drug reaction or adverse effect. So pharmacovigilance is study of adverse effects of the drug. Study of adverse effects of the drug is called as pharmacovigilance, and we do 4 things in pharmacovigilance which are remembered as DAUP DOP. What are the 4 things? We do detection, assessment, understanding, and prevention. So detection, assessment, understanding, and prevention of adverse effects of drugs is pharmacovigilance. So one by one we will discuss. First of all, we will detect. First of all, we will detect whether there is any adverse event or not. So as we discussed earlier, there are two terms. One is adverse event, second is adverse drug reaction. Now, what do you mean by adverse event is anything which happens which is wrong, which is not wanted while the person is taking a drug. Okay? So we have started a drug to a person, and whatever wrong happened to that, we will start noting. Everything will be adverse event. Like we write the person developed vomiting, it is an adverse event. We write the person becomes sleepy, it is an adverse event. The person falls and he develops fracture, that is an adverse event. Person's girlfriend leaves him, that is an adverse event. So adverse event is anything wrong which is happening. We do not think whether the drug is causing or not. Whatever wrong is happening, we will write that. Okay. Now, out of the adverse events, we want to know which are caused by a drug, which are not caused by a drug. So if it is caused by a drug, then it is called as adverse drug reaction. Okay. So that means all ADRs are adverse events, but all adverse events may not be ADRs because many are not caused by it. Like if a person develops vomiting, most likely it will be adverse drug reaction. Like if a person was going and person falls and develop fracture, can it be adverse drug reaction? Yes, it can be, because the drug may be sedative. The person lost his balance, the person falls. That can be adverse drug reaction. Okay? That may not be adverse drug reaction, may be unrelated to the drug. The girlfriend of the person has left him. Can that be ADA? Yes, it can be ADA. The drug may have caused impotence. That may have resulted in this thing. So adverse drug reaction is when it is caused by a drug. Okay. So in first thing we will do detect whatever adverse events the drug is causing, we will detect. Simply we will write it down. Okay. Then second, what we will do is we will assess. We will assess two things. First, we will assess which adverse events are caused by drugs. So that means which are ADR, which are not ADR. So we will do causality assessment. Causality assessment. That means we will want to know whether the drug is causing or not. And second thing we can do is severity assessment, how severe the adverse drug reactions are. Okay. Now in causality assessment, there is one scale known as Naranjo scale. So basically we have a questionnaire on which we will keep on writing whether this is true, this is true, this is true or not. So finally we will come to know that whether it is caused by a drug, likely to be caused by a drug or not. Okay. One of the simplest methods of assessing the causality you can see is a D challenge and re-challenge method. D challenge and re-challenge. Okay. You can simply know that, like the person was developing vomiting. So what we will do, we will stop the drug. We'll stop the drug. Now the person is not having any vomiting. Okay. So after a few days we start the drug again the person will vomiting. So that means it must be caused by a drug. Okay. So D challenge means stop the drug and re-challenge means restart the drug. So that is one method of causality assessment. Okay. Then severity, we will see whether the adverse effects are very severe, life-threatening, or they are mild to moderate side effects. Okay. Assessment. So after doing the assessment, we will try to understand. So we will now apply our knowledge of pharmacology and knowledge of medicine to understand what is the mechanism, how the drug is causing side effect. Okay? Like in NSAIDs are causing peptic ulcer. So we apply our knowledge of pharmacology. We came to know that NSAIDs are decreasing the prostaglandins. So the prostaglandins were protecting the stomach. So protection has gone, that can lead to peptic ulcer. Okay? So then we will try to prevent these adverse effects. Like in case of NSAIDs, we will advise that take them after the food. Do not take them empty stomach. More chance that they will cause peptic ulcer. Okay? So these four things: detection, assessment, understanding, and prevention of adverse effects, we will do in pharmacovigilance. Okay. Now, pharmacovigilance, India has made a special program which is known as National Pharmacovigilance Program of India. National Pharmacovigilance Program of India. Now, what is done in this program is almost all the hospitals, almost all the private hospitals, they have been given the work to collect the adverse effects of the drugs. Okay? So these are called as peripheral centers, also named as adverse drug reaction monitoring centers. Adverse drug reaction monitoring centers, or AMCs. So almost every medical college, every big hospital, they have been given a room. In that room, they have been given a computer and a software. The name of that software is Vigiflow. Vigiflow. So whenever any person comes with adverse effect, they will enter the data in that. Okay? So Vigiflow will collect all the data from all the ADR monitoring centers. Okay? Then all the ADR monitoring centers, they will go and report it to the National Coordinating Center. National Coordinating Center. National Coordinating Center, which is the center of India. It was previously present in AIMS, but now it has been shifted to National Pharmacopoeia Commission at Ghaziabad. In UP, it has been shifted National Pharmacopoeia Commission. Okay? So similar to India, every country has National Coordinating Center. So all the National Coordinating Center of different countries, they will report to Uppsala Monitoring Center, which is present in Sweden. Uppsala Monitoring Center or UMC, it is present in Sweden. So Sweden get all the data. Okay. So Uppsala in the Sweden, it will get data from all the countries. So National Coordinating Center of all different countries, they will report to Uppsala. Okay. Now this Uppsala is in direct touch with the Food and Drug Administration, FDA. So they are analyzing the data that this drug is causing this side effect. So many countries have reported. So they will report it to FDA. So FDA will analyze that data and then they will give the guidelines. Like sometimes the FDA can give a black box warning. Like if a drug is causing some side effect which was not earlier reported, now the FDA will ask the manufacturers of that drug on the label, write the warning in the black box that this drug can cause this side effect. Okay? Or if the side effect is seemed to be very, very severe or very harmful, then the FDA can give the ruling that withdraw the drug. They can ban the drug also. Okay? So these different things can happen. So quickly seeing what happens, the architecture of National Pharmacovigilance Program of India is almost all the hospitals, big hospitals, private hospitals, government hospitals, medical college, they have been called as ADR monitoring centers. They have been given a software called as Vigiflow. They will enter the data of the adverse effects. So that data will be sent to the National Coordinating Center, which is at National Pharmacopoeia Commission now, Ghaziabad, UP. Previously it was at AIMS. Now it is at NPC Ghaziabad. Now this coordinating center will send the data to Sweden, Uppsala Monitoring Center. All other countries will also send there. So data will be analyzed and then that will give the information to FDA and FDA will give the guidelines what to do. Okay. So that is National Pharmacovigilance Program of India. Okay. So these are the important things we discussed in the chapter. So quickly revising what you should know about this chapter, particularly in the beginning, we discussed the clinical trials, 4 phases of the clinical trials, phase 1, 2, 3, 4. The important points we discussed, which is the first phase in humans, which contains maximum number of patients, what is done in which phase, that you should know. Then phase 0, we discussed the separate thing. You should know about phase 0 clinical trials. You should also know about the blinding, which are single blind, double blind studies, what is placebo, all of these you should know. And you should know the adverse effects, event versus adverse drug reaction, what is the difference, you should know. Various types of adverse drug reactions, type A, B, C, you should know about them. And lastly, you should know about pharmacovigilance also. Okay, so all these are important for MCQs. So now we can quickly discuss some MCQs. Which of the following statement is correct about clinical trials? You need to tell correct. New drug application is filed with phase 2 clinical trials, right or wrong? Wrong. New drug application is filed after phase 3. Okay. Phase 0 clinical trial is for checking the pharmacokinetics of a drug. Yes, this is right. We know that in phase 0 we cannot know efficacy, we cannot know safety. Only we are monitoring on the counter. In phase 3, open label studies are done. Wrong. In phase 3, we do double blind studies. Open label are done in phase 1. Okay. Phase 2 is post marketing surveillance. This is wrong. Post marketing surveillance is phase 4. Okay. New drug application is filed after which phase? NDA is filed after which phase? Yes, it is filed after phase 3 when the drug is ready for marketing. Okay. A study was done to know the efficacy of a new drug. Some subjects are given new drug and some subjects were given placebo. Neither subjects nor investigators know who is receiving a drug and who is receiving placebo. So which type of study it is? Yes, this is double-blind studies. Such studies are conducted in which phase? Double-blind are conducted in which phase? Yes, it is in phase 3. A new drug is introduced in the market after, read very carefully, after which phase? It is after phase 3. Okay. After phase 3, the drug will be marketed. Manufacturer of a drug company labels the drug contains 500 milligram paracetamol. On quantitative analysis, authorities want to contain 200 milligram. So the drug contained is lesser. So according to Drug and Cosmetic Act, which type of drug it is? So it is wrongly labeling the drug. So it is misbranded drug. Misbranded drug. Okay. Identify the true statement regarding clinical trials. Again, true. Phase 1 is done to determine efficacy. Wrong. In phase 1, we cannot know the efficacy because it is done in healthy people. Healthy volunteers are recruited for first time in phase 2 is wrong. Patients are first time recruited in phase 2. Healthy volunteers were done in phase 1. Randomized control trials in patient done in phase 3. Yes, it is randomized double blind control studies. Phase 4 is pharmacokinetic study in animals. Wrong. Animals it itself wrong and pharmacokinetic is not phase 4. Okay. So the answer here is C. Okay. So these are the important points regarding the clinical trials and pharmacovigilance. My take home message is great things never came from comfort zones. So you have to move out of your comfort zone, only then you can become great. Okay? Thank you very much.